Harmony. Harmony clusters all the cells in the PCA space. So I'm going to come back very briefly here on fast MNN. So fast MNN does not work in the PCA space, but it really works in the gene expression space, which means that every axis here in this space represents one gene for which the expression was measured. So this is a very high dimensional space that we're working in, because we typically have 20,000, 30,000 genes that were measured in our experiments. Because this is such a high dimensional space, it also means that it will take some time to do the calculations here. It's more complex. And it also requires more resources from your computer to do the calculations. So how does Harmony work? Harmony will cluster all the cells in the PCA space. So all the cells from all the samples together. So it works in the PCA space and not in the gene expression space. So the PCA space is typically 30 to 50 dimensions, which means it is much smaller and much less complex than the gene expression space. So Harmony will be a lot faster than fast MNN. So what it will do is it will make one big clustering for all the cells from the 3 different samples here in this example. And it will look for clusters that are a mixture of cells coming from different samples. So cluster 2 is the best one here because it contains cells from the 3 different samples. Cluster 1, cluster 3, and cluster 4 contains cells from 2 different samples, as you can see here. And what he will do is, for every cluster, he will look at the cells that belong to the same sample, and he will calculate the center of that population. So the center of the cells in cluster 1 that belong to the green sample is located here. The center of the cells in cluster 1 that belong to the purple sample is located here. And he will do the same thing for the other clusters. So you will get centers located here in the PCA space. And what he's going to do then is, for every cluster, he's going to move these centers closer to each other, as you can see here. And he's going to move all the cells along. And in this way, he is doing the batch correction. This is less harsh than the Seurat batch corrections. When do you use this one? Well, for the small batch effect. So here, batch effects between replicates, batch effects between patients.